electrolytes here. All right, just quickly here, let me just make a note of who is on here. Oh, there's Alex. There's no Alex today.
I think she's on a volleyball game.
Oh, she might be. I think you're right about that. Okay, so we were talking about thermoregulation, how the body maintains its body temperature, right? And most of the time, our issue is that we have excess heat to get rid of. You know, all of our cells produce heat. When ATP is formed, 60% of that energy just becomes heat. Even when ATP is used, heat is always released as part of that process too. So, you know, we're like giant furnaces. So we always have heat to get rid of so we don't get hotter and hotter and hotter. So when the body temperature starts to rise, the heat loss center is activated. So thermoregulation is controlled by the hypothalamus. So when the temperature in the hypothalamus goes up or down, it responds with either heat loss mechanisms, in other words, get rid of heat, or heat gain mechanisms, you know, conserve heat and generate heat. So when we are too warm, you know, multiple things happen. One is behavioral changes. Our posture, or how our bodies are positioned, plays a major role in how much heat we radiate. So when we're cold, we tend to bolt our arms and legs in, and we tend to flex. You know, we hunch over to keep our skin next to our skin. When we're warm, we do just the opposite. You know, we want to extend our arms and legs. We want to get our arms and legs out away from our bodies so that we can more effectively radiate heat. Other kinds of behavioral changes, you know, if you are overdressed for the weather, you know, and you go outside and you start to get warm, eventually you take your coat off, right, because you've gotten hot. That's a behavioral change too. And the truth is, those behavioral changes are very hard to resist or prevent. You know, so, you know, if you were in a winter coat in a warm room, it would be very difficult for you to leave that coat on to become heat exhausted. You know, you'll take it off before you'll do yourself harm, essentially. So that's one, behavior changes. Another is the skin. The skin is our radiator, right? So when we have excess heat to get rid of, blood goes to the skin because the skin sits right next to the air, and the air is usually cooler than our body temperature. Now, in the middle of summer, not always the case. But outside of that narrow time frame, most of the time the air around us is cooler. So if we put our warm blood next to that cool air, we lose heat from radiation and convection. So blood is shunted to the skin. And sweat production. This is a human trick. Most animals can't do the sweat on the skin business. But what we do is we put small amounts of water, we secrete small amounts of water on our skin. As that water evaporates, heat is taken away with it. Anytime water evaporates, whatever surface it left becomes cooler in that process. So we put blood near the skin, and we put sweat on the skin, and together we get this cooling system. And then finally, we have a respiratory system for getting rid of heat, too. You know, the lungs are essentially thin membranes, thin, wet membranes with air next to them. So as we breathe, we evaporate moisture. As we evaporate moisture, that lowers the temperature, right? Because it's evaporative cooling again. So when we're hot, when we're warm, we'll breathe deeper and faster, and we'll have a tendency to breathe through our mouths instead of our nose. Because when we breathe through our mouths, that water can actually leave, you know, so the evaporation takes that water takes the heat away with it. So the reason your dog or your cat pants in the summertime or when they've been running is they're using their respiratory cooling system. By panting, moving air in and out all the time, evaporation occurs and it cools the inside body temperature. So it helps them to control their body temperature because they don't have sweat glands on their skin to do that role. So they only have that one. So 4 mechanisms for heat loss, getting rid of excess heat. All right. Well, what about the opposite? there are times, winter does happen on Earth, and when the air temperature gets very, very low, we have to make adjustments to our heat conservation system in order to accommodate that. So when the hypothalamus gets too cool, we're going to see these responses, heat gain responses. So they're basically two kinds. There's conservation and there's thermogenesis. If we're too cold, we want to hold on to every degree, so to speak, right? So we don't want to lose the heat that we're producing. So some conservation changes, it's the opposite of what we see in heat loss. And that is when we're cold, the blood moves away from the skin. Because if we can keep blood away from the skin, we'll hold on to our body temperature better because there is none of that radiation. So when you studied the vessels for the last chapter, you saw that in the arms and legs, there are superficial veins and there are deep veins. And here's the reason why. Because when we're warm, blood travels through the superficial veins, right? But when we're cold, blood travels back through the deep veins. So that heat is conserved and not lost to the environment through the skin. And then another trick that evolved is the deep veins and the deep arteries are bound together. They're wrapped together, kind of like you would wrap cords together. And the reason for that is to allow for heat exchange. So as warm blood is coming from the heart through the artery, here we have 37-degree blood coming in, right? So this is the artery going out. This is the vein going back. Because the two are right next to each other, as the blood is moving, the heat is transferred directly from one to the other. So by the time you get out here to the hand, the blood temperature arriving at the hand is low, so 24 degrees, much, much lower. So that heat is conserved up in the arm. It's one of the reasons why your hands are the first things that get frostbit or go numb or start to hurt in the cold. It's because the blood that's getting to your hands is literally colder than the blood that's in your chest because of this heat transfer mechanism. It's useful, though, because the hand can tolerate cold much better than the liver can or the heart can. So, and remember, for us humans, we get to wear our big warm boots and our feet never touch the snow. But in your cats and dogs, again, this system is critical because otherwise they would lose an immense amount of heat through their paws on the ground. So it helps to hold on to that heat transfer mechanism. All right. So those are conservation. We haven't made any heat, right? But we have kept us from losing it. But we also have two systems for creating heat, creating more heat when we need it. And that is shivering and non-shivering thermogenesis. So genesis is to make, thermo is heat, thermogenesis. So we'll start with the shivering one because that one we've all experienced. So when it's cold outside, to create extra heat, the body increases muscle contraction. We call that muscle tone. Muscles are always contracted a little bit. Well, when your body temperature starts to fall, the muscle tone increases. So when it's 20 below outside and you go and the car's not warm and you get very stiff, right? You're all huddled up and your muscles are contracted. That is shivering thermogenesis. Contract the skeletal muscle because contracting the skeletal muscle creates heat. Now, if that muscle tension isn't enough to create the temperature that we need, then we get into the oscillations where the muscles relax and contract and relax and contract. And by doing so, they create even more heat. So that is true shivering. But the stiffness you feel when you're outside in the cold, that is also shivering thermogenesis because you're on your way to start shivering. You just haven't done that yet. So this shivering, this muscle contraction can generate 400% more heat than normal from a single skeletal muscle. So it's very, very effective. Okay, so that's shivering. And then also the adrenal gland releases epinephrine in response to low body temperature. And epinephrine increases metabolic rate all over the body. So all the cells of the body start burning more fuel, and as they burn more fuel, they create more heat. So while the muscles are contracting to make temperature or to make heat, all the other cells are increasing their rates of metabolism to do the same thing. So one thing that people will notice is if you are outside a lot when it's cold, your caloric needs actually go up because some of the food that you eat isn't becoming metabolism and it isn't becoming muscle contraction. It's becoming warmth. Basically, you're keeping yourself warm by burning through calories. So like the scientists that work down in the Antarctic, they sometimes have 3,000 or 4,000 calorie diets because they're outside so much having to generate all this heat. So heat gain center, conservation, and generation of heat. Okay, so that was the last topic. But let's do a question here real quick. No, we're going to skip that one. This one we are going to do though. Okay. The vitamin essential for blood clotting is which of those? Boom, there we go. Very good. Vitamin K. That is the correct answer. The only other I wanted you to know about vitamins. A, D, E, and K, ADEC, right? Those are the fat-soluble vitamins. Vitamin D is involved in calcium homeostasis, and vitamin K in clotting. I didn't ask you to know anything about A or E. And then in the water-soluble vitamins, those are all the B vitamins. And I wanted you to know that you have to have them as a regular part of your diet because they're not stored anywhere. All right. Okay. In the interest of time, we're going to move to the review. All right. And I have posted this already, so feel free to use my lists here. All right. So review points. What do I mean by that? I mean these are things that in your studying you should be sure that you have looked at because they're important points. Certainly, like was said earlier, I can't go through everything that we talked about, but these are the things that come to my mind as we go through each of the chapters. Okay, so lymphoid system and immune system. I think of that chapter in two parts: the lymphatic vessels and organs. That's one piece, and then the immune system is the other piece. So we learned that the lymphatics. I'm actually going to do it this way because it's faster. The lymphatics are this system of vessels that originate in the tissues, and they come back together or come back into the trunk. Their job is to bring excess fluid from the tissues back into the cardiovascular system, right? So like lymphatic capillaries start as sort of blind sacs. They form vessels. These vessels come together, and eventually they dump into the cardiovascular system in two places. Okay, so one is at the thoracic duct. That's right here. So the well, here I'll just draw it as a picture. So we have here's a person, right? So we have 1/4, 3/4, right? So all of this here, so left side of the head, left arm, whole lower extremity, that's all thoracic duct. Over here in our picture, it's right here. And then this quarter of the body, so the right side of the head, the right arm, and the right upper chest and back, that comes in through the right lymphatic duct. Okay, so 1/4, 3/4, right lymphatic duct, thoracic duct. All right.
I feel like we went through that like a year ago.
Yeah, I know. It seems like a long time. And then we talked about the lymphoid organs and tissues. The lymphoid tissues we talked about, that was the tonsils. Remember, there's 3 sets of tonsils. The MALT, mucosa-associated lymphoid tissue, the lymphoid cells that are found in the mucosa of the GI tract and the respiratory tract. So those are the lymphoid tissues we talked about. The lymphoid organs, that's the spleen, the lymph nodes, and the thymus. So be sure you look at the, what those things do, lymph nodes, spleen, and thymus, and, sort of what they're for. And then we moved into the immune system, and we talked about the nonspecific defenses, which are listed here, and we're not going to go over each of them again, but be sure you do. So go through each of the nonspecific defenses. You should know for sure that once the physical barriers have been breached, the first line of defense, of cellular defense, is the phagocytes. Those are the cells that a would-be invader encounters first. All right. And let's see. Okay, so the nonspecific defenses. Remember that we had two branches to the immune system, acquired and innate. The acquired system is the T cells and B cells, and the innate system is the neutrophils and the eosinophils and basophils and macrophages. Okay, so when you hear acquired or I have those reversed. Acquired goes with specific, and innate goes with nonspecific. Sorry, that's my error. But what I was going to say is, when you hear acquired, think B cells and T cells, because that's what runs that system. All right. We talked about antigens, right? An antigen is a piece of a thing. These immune system cells, they cannot see, so the way they tell things apart is by looking at parts of a thing, So you could potentially identify a shoe from just a little part of its, sole, right? So that's the idea of an antigen. Is you can identify a whole thing from a piece of a thing. And both T cells and B cells work with antigens. They have one antigen that they're looking for, and when they see or find that antigen, then they activate or move to the next step of activation. All right. T cells and B cells. T cells are born in the bone marrow, but they arrive at they go to the thymus for training, and we have two types. We have TC cells and TH cells. So these are cytotoxic. They kill cells, right? So cytotoxic T cells are important in destroying virus-infected cells and mutated cells. TH cells are T helper cells, and they have two basic roles. One is to activate B cells, and the other is just to help. So they coordinate the other actions of the immune system. So they're like manager cells, so to speak. So those are the T cells. And then the B cells are called B cells because they're born in the bone marrow. And when they're activated, they become plasma cells, which make antibodies. And an important word, another word for antibody is immunoglobulin. In your book, this is the word your book uses. I call them antibodies. It calls them immunoglobulins. Okay, so this equals an antibody. So we'll just say, because in your notes you can say antibody, right? So B cells are activated when they see their antigen. They're turned on by helper T cells. Boom, they become plasma cells, which are antibody factories. And antibodies are immunoglobulins. Okay. There were 5 types of immunoglobulin, of antibody. You should review the claim to fame for each one, right? Because there was IgA, IgG, IgD, IgM, and IgE, right? So look at those. Okay. MHC molecules, complex topic, super complex topic, really, MHCs. But the short version, what I want you to know about them, is where they are and what they do. So we'll say MHC. Class 1s all nucleated cells, right? So all cells have class 1 MHCs. Class 2, they are found only on immune system cells. So class 1 MHCs, they work, they're all about virus and mutation. So when a class 1 MHC has an antigen inside, it's assigned for the immune system to destroy that cell. So the TC cells, you see that, cytotoxic T cells, they work with these class 1s. The T helper cells and B cells, they work with these class 2s. All right, so we have our two different types, so class 1s and class 2s. So class 1s, all cells, class 2s, immune cells. That's really the take-home message. Okay. Our T cells have a fancy name that relates to how they, to HIV, which we didn't get into that, but you will later. So T helper cells are also called CD4 cells. Cytotoxic T cells are also called CD8 cells, and we talked about that too. The CD4s work with class 2s, and the CD8s work with class 1s. So a mnemonic is multiply the class and the CD count always equal 8. So like 4 times 2 is 8, and 8 times 1 is 8. So they help those two things go together. Okay. All right, T cells, about that. The immune response, primary versus secondary, that was about antibodies. The first time your body encounters a pathogen, it makes a little bit of antibodies, but the second time, it makes a lot of antibodies, right? So you have this big old curve. So the secondary response is faster and more vigorous than the primary response. This is why we don't get the same virus twice, is because the first time we have to build an attack, right? The second time we have an attack already planned. So a lot of times we can be exposed to a virus like chickenpox and not know we've been exposed the second time because it couldn't make an infection because there were already antibodies present. And the immune system pieces, the acquired versus the innate, they don't work separately, they actually work together. And probably the biggest distinction between how they work is the pathogen type. So bacteria typically invade outside of cells, right? Viruses and cancer invade from inside of cells. So we have sort of two different pathways. So be sure you review these differences. So like for bacteria, it all begins with phagocytosis, right? The bacteria are swallowed and destroyed. Their pieces are presented on the membrane. That's antigen presentation. That turns on the acquired system. So the cytotoxic T cells are killing the bacteria. The helper T cells are getting everything else up and running. They activate B cells, which make antibodies attached to the bacteria and help to destroy them, right? The virus pathway is more challenging because the immune system can't get to the virus. You know, immune system cells can't crawl inside other cells. So they have to behave differently. The way that a viral infection is handled is the virus infects, it creates antigens on the surface in those class 1 MHCs. That activates the T cells, cytotoxic T cells, and infected cells are destroyed, right? So, because you can always replace a cell, but a viral infected cell is a virus factory. So the infected cells are killed, interferons are released, that helps other cells not get infected, and then eventually B cells are activated, made into, and they make antibodies, which then aid the immune system in identifying and destroying the virus. So sort of two different pathways depending on whether it's a bacteria or something else. All right, so boom, was the first chapter that we covered this unit. All right. Shifting gears, respiratory anatomy. We talked about the nasal cavity, all the way to the alveoli. The nasal cavity warms and humidifies. Know what the function of the nasal cavity is, because that is for sure on the exam. And then we followed the airways all the way down to the alveoli. So we had our, that was this, right? We went from nasal cavity to pharynx, pharynx to larynx. We talked about the glottis, right? The glottis opens and closes, guards the lungs. Also is where the voice box is. We looked at that creepy movie of the lady singing, right? We looked at the trachea. That just goes straight down. The trachea branches. We have our bronchial tree. Just like we made an analogy to a tree outside. Where as you go up, the branches get smaller, but there gets to be more of them, right? Until eventually you get all the way to the leaves. And the leaves, in our case, are the alveoli. Those little sacs of air, essentially, here. And the alveoli are surrounded by capillaries. So we have between air here and blood here, we have basically three very thin pieces, right? We have a type 1 pneumocyte here. We have an endothelial cell. That makes up the capillary wall. And then between them, we have a basement membrane, or we'll call it a basal lamina. So all three of these things are the respiratory membrane. That should ring a bell, I hope. If it doesn't, be sure to mark it. So respiratory membrane is what's between air and blood. And we've got type 1 pneumocyte. That's what makes up the wall of the alveoli, right? Very thin. And then we have our endothelial cell, very thin, and we have the basement membrane in between. So CO2 is going to move from blood to air. Oxygen is going to move from air to blood across this membrane. Know also that the type 2 pneumocytes here, they make surfactant. Surfactant helps keep the bubbles open, right? Helps keep the alveoli open. And then we have our alveolar macrophages that help keep everything clean. They're like robot vacuum cleaners. All right, so here's that respiratory membrane: air, capillary, and the three things in between, right? Okay. Let me go back to my list. Okay, so respiratory membrane, types of pneumocytes, we did that. Pulmonary and alveolar ventilation, we can skip that because in dead space. Gas exchange. All right, so across this respiratory membrane, right, we have the movement of carbon dioxide and oxygen, right? So we have, CO2 is going this way, oxygen is going this way. And both are moving by diffusion, right? They're moving from high partial pressure to low partial pressure, or vice versa. I think, let me see if I have that slide. Yes, I do. Okay, so maybe this will look familiar. So alveolus, blood coming into the lungs, blood leaving the lungs. These are representations of tissues of the body. Arterial blood coming in, venous blood going out. So we see the partial pressure, which is a kind of concentration. How much in how much volume, right? So the numbers, or the movement, is always from high numbers to low numbers. So, 100 to 40, CO2, 45 and 40, so it's going to go the other way. So review the numbers here and review the directions that things are moving in, right? So the blood that has the highest concentration of CO2 would be found, here, leaving the tissues, or here, arriving at the lungs, because essentially this is the same blood, right? What leaves the tissues goes back to the lungs. All right. Then, okay, so that's gas exchange. Oxygen transport. We know that most of the oxygen that's carried in the blood is carried by hemoglobin. And we looked at this curve, which undoubtedly was a little confusing, so we'll look at it just one more time. All right, so what hemoglobin needs to do is it needs to gather up oxygen where there's lots of oxygen, and it needs to release oxygen where there is very little oxygen, right? So we can't have a molecule that stays the same no matter what. We need to have a change. And what this curve shows us is when there is a lot of oxygen around, so this is the amount of oxygen along the x-axis, and this is how much oxygen hemoglobin is carrying, right? So the more oxygen there is, the more oxygen hemoglobin carries. That is the easy part. So where there's lots of oxygen, hemoglobin gets saturated. In other words, it gets full. But as the oxygen level goes down around the hemoglobin, what we see is hemoglobin starts releasing oxygen. So the difference between this point and this point is how much oxygen was released. So the hemoglobin's trick here is it gathers oxygen where there's a lot, and it dumps it where there's less. And then when the pH is low or the temperature is high, this effect is even more pronounced. So in a busy tissue, hemoglobin releases even more oxygen. Okay? And virtually all of the oxygen that is carried around the body is carried as by hemoglobin, as we see here. So oxygen comes in, gets bound to hemoglobin. In the tissues, hemoglobin releases the oxygen, and it goes into the cells. And then, so that's oxygen delivery. But now, how do we get the CO2 back? And we had some important numbers there. Okay, so 7% of the CO2 goes back as PCO2. 23% is bound to hemoglobin. And then 70% is as bicarbonate. And that was because of this equation, which ought to look familiar to you. H plus bicarbonate. So in the tissues, it goes this way. Tissues. And then in the lungs, it goes this way. So essentially, we can transform CO2 into bicarbonate and carry it that way. So 70% of that is from that form. I actually have a different slide up here. So that's this slide, the 7% here, 23% here, 70% here. Now, this bit right here is important. So the bicarbonate gets made inside red blood cells, but it has to get moved out of the red blood cells, or else this equation will start to back up. So what we see here is in the tissues, bicarb is pumped out of the cell, and a chloride is pumped in its place. And then we also see that this H plus gets carried by hemoglobin. So this goes up here to be carried back to the lungs, and this goes out. So it goes out of the cell. Because when we get to the lungs, the whole thing reverses. So if this slide doesn't look familiar to you, it should. So have spent time looking at this one, because this is there's a lot of information in a very short one picture here. So the top is oxygen carrying. The bottom is all this CO2 stuff, which is the more complicated part. So just like we were talking about, CO2 gets added to water to make carbonic acid, which breaks down into bicarb. Bicarb is shifted out. The H plus binds to the hemoglobin, right? Then in the lungs, that H plus is pulled off. The bicarb is pumped in. Carbonic acid is remade, but now it breaks down into CO2 and water, and then the CO2 exits. So that's the 70%. This is the 23% here, and then the 7% is out here. All right? So that's carbon dioxide transport, and in terms of clinical relevance, hugely so. When you stop breathing, one of your principal problems is that your hydrogen ion concentration goes up because you're no longer getting rid of this CO2. So this system backs up. We get more and more hydrogen ion. We get more and more acidotic, and our respiratory drive fails. And then, the brain goes into coma, and now you're circling the drain. So this respiratory stuff is important because it can kill you real fast. Okay. And then, let's see what other topics. All right, so gas exchange, oxygen transport, CO2 transport. Okay. A little bit about breathing mechanics. How do we breathe? Okay, this is how the molecules move around. But how do we actually move air around? And we do that by changing the volume of the chest. So we had, let me go back to that slide because it's pretty small. All right. So we have to do two things, right? We have to get air in and we have to get air out. So the way we do both of those things is by changing the shape of the chest. So when we inhale, the diaphragm contracts, so the bottom goes down. The ribs lift up and out, so the walls go out, so the volume of the chest increases. When you take a thing filled with gas or air and you increase its volume, the pressure goes down. So we increase the volume to lower the pressure until the pressure inside the lungs is greater than the pressure out here. That causes air to flow in, right? So that's inhalation. Expand the chest, bring air in. Now, exhalation is the opposite. The volume of the chest decreases. That increases the pressure. When the pressure inside is greater than the pressure outside, air goes out. Okay, so breathing mechanics. It's all about changing the volume of the chest. So know which is which. Expanding the chest is inhalation. Contracting the chest is exhalation. And you can use yourself as a model there. You can feel that your chest gets bigger when you inhale and smaller when you exhale. All right. There are muscles that do that. So I did want you to know the respiratory muscles, both the primaries and the accessories, because here again, in respiratory failure, when you do your assessment of a patient in respiratory distress, you're going to look to see if they're using these extra muscles at rest. Because normally, a patient comfortable at rest is not using these accessory muscles, right? That says there's something wrong. They're working harder to breathe than they should. So the primary ones, external intercostal and diaphragm. Those are our primary breathing muscles. So we use these all the time because inhalation is active, exhalation is passive at rest. So the external intercostals lift the ribs, the diaphragm pulls the bottom down, so air goes in. These other muscles, accessory inspiratory and accessory expiratory, are used when we're breathing hard or when we're breathing against resistance, like when we're talking or playing an instrument or something like that. So review those muscles. Most of them are muscles that you learned last semester anyway. The most of the expiratory muscles, three of the five here, are abdominal muscles, which I know you learned last semester. All right. So muscles of respiration, breathing mechanics, we did that. Lung volumes. I'm only going to go through one in the interest of time here. All right, so we looked at this graph. So here's our tidal volume, right? You know, we're breathing in, breathing out. Well, if we take the biggest breath in that we possibly can, and we blow as much of that air back out as we possibly can, the amount of air that we have moved, that's called the vital capacity. Did you guys do that in lab? You did. Okay, so of all those measures, the one that should stick in your head the best, well, two. One is tidal volume. You know, we're breathing all the time. The other is vital capacity, because that is the maximum your respiratory system can perform. Okay, so, you know, vital capacity doesn't change, like at least not over the short term. These others can. So vital capacity is biggest breath in to most smallest or biggest breath out. Vital capacity. All right. Lung volume. And then respiratory control. Okay, so the control of respiration is CO2. Well, here, let me back up. Okay, so we have CO2 and we have the pH. They're inversely related. What that means is as the PCO2 goes up, the pH goes down. So the more CO2 in the blood, the more acidic we become. And so the brain decides how much to breathe by looking at this, which tells it about this. So what is the primary control of respiration? For our purposes in the respiratory system, we're going to say it's CO2. What we're going to see in the acid-base system is it's actually pH. But there won't be any questions that have more than one answer, I hope, anyway. So for our purposes, CO2, that is the primary thing that controls respiration, okay? Because I know that I may have confused that issue in class because there is a pH component, but for our purposes for this exam, PCO2 is the controller for respiration. All right. And I can see already that I'm not going to get through all this, but I have an answer. In the other section, in my other A&P section, we did get through all the review points. So I'm going to copy that Tegrity over to our Tegrity site so you have it, because I don't think I'm going to get to this, because I only have 8 minutes left. Okay, the digestive system. We followed the whole thing from mouth to anus, right? Classic way of studying the digestive tract. So we looked at the mouth and we looked at teeth. All I want you to know about teeth is there's a picture in your slide. The slides look a little bit like this, where there's a tooth on top and then there's some labels, right? That's the one I want you to look at. So, you know, there's enamel, that's the hard stuff, right? There's the pulp cavity, and then there's cementum along the root of the tooth. So just review that quickly. Most of it is stuff you probably learned in high school in reality, but do look at the picture that looks a little like that. Okay, we talked about saliva. Remember, carbohydrate digestion starts in the mouth. Yes?
Notice that on the end of this, you have a whole bunch of questions. Do you have the answers anymore?
Yes. And I have to post it in 2 pieces because there's not an easy way to put the answers in the PowerPoint. So there'll be this online, and then under it, there'll be the questions and answers. They're the same questions that are on the PowerPoint. They're just, you have the answers at the end, because that's how the software works. All right, so salivary glands, pharynx. Swallowing, be sure you review that. You know, there's 3 phases to it. Review the parts, know what the epiglottis is, and know that the pharyngeal phase happens very quickly. You know, we saw that in that picture, how it would go past the pharynx. Know for the stomach, know the phases, so cephalic, gastric, intestinal. Know the different cell types. So G cells, parietal cells, chief cells, know what they secrete is what I mean. So G cells make gastrin, parietal cells make acid, and chief cells make pepsin. All right? Duodenum, know that that's where the pancreas and liver secretions go in, and review the secretions of both, the pancreas and the liver. Know the specializations of the jejunum and ileum. Remember we talked about surface area expansion. The jejunum has deep folds, plica circularis, right? Large villi. The ileum less so. The duodenum least so. Because the more surface area, the better the absorption and digestion. And then for sure, know the large intestine anatomy and what it does. The large intestine compacts feces. That's one way to put it. Another is it removes water and salt from the feces. Same idea. Make it smaller, make it drier, is what the large intestine does. And know that up, across, down, around, and out. All right. The TCA cycle, all I want you to know really is that short version that we went through. Okay? You know, so the carbon dioxide is formed in the citric acid cycle. And then the electron transport system uses hydrogen ions to make ATP. That's pretty much the extent of what I want you to know there. Do spend some time on this bit. In that metabolism chapter, we go for each of the nutrient types, protein, lipid, carbohydrate. We follow from ingestion to metabolism. And you should go through those steps. You know, so you should know that fat digestion and carbohydrate digestion start in the mouth. Protein digestion doesn't start until you get to the stomach, right? And then the caveats or the weirdnesses of lipid digestion, that lipids don't end up in the bloodstream first. They end up in the lacteals first, in the lymphatics. And then they have that chylomicron, LDL, HDL. So review what those words mean. Okay, so chylomicron, LDL, HDL. Look at absorptive and post-absorptive states. Compare the two so that you know what's happening. Essentially, in the absorptive state, the short version is glucose is used by cells all over the body. In the post-absorptive state, glucose is spared except for the brain. And the liver releases glucose and makes glucose instead. Because we're trying to keep those things, those levels high. And then all the ways to feed a mitochondria. Okay, so we feed a mitochondria carbohydrates by breaking glucose in half, right, to 2-3 carbon chains that we feed them. We call that glycolysis. It happens in the cytoplasm. Makes 2 ATP, right? Every molecule of glucose yields 36 ATP. And then the other two things. Well, what about proteins and lipids? Before proteins can be fed into the mitochondria, we have to strip the nitrogen off first. So we call that deamination, right? And that makes an ammonia, which is toxic. So that ammonia gets made into a urea, right? Which is not toxic. And then to burn a fat, we use beta oxidation. So we clip off 2 carbons at a time, feed them to the mitochondria, and we get a boatload of ATP back out. And then amino acids. Most amino acids we can make ourselves. The liver can do it. We talked about 2 tricks for that. Amination, adding an ammonia to a carbohydrate. The other was transamination, where we have 2 molecules and we flip the nitrogen from one to another. And all those things are to keep the amino acid levels where they should be. All right, A, D, E, and K, that's just the vitamin thing again. And then thermoregulation we just talked about today. All right, so none of the slides in this slide set are new. They're all snatched from other places. I just stick them in here so I have visual aids to use when we review stuff. I'm just going through to be sure we didn't miss something. And then we get to a bunch of questions. All right? So that is it for today. Study hard. I know it seems like a lot, but you'll be, if you stay organized in your study, you'll do just fine. Use the PowerPoints as your guide. And I will still move that integrity over because we had more time, so I got to talk more about something.